Good afternoon, everybody. And so this is a map of India, and I came from this province called as Uttar Pradesh. And this is Lucknow, where I did my PhD at CSIR National Botanical Research Institute in 2012. And then I moved to the 1 year for my postdoc at International Center for Genetic Engineering and Biotechnology, New Delhi. It is a United Nations organization. And then in 2013, August, I came to this university, Western Kentucky University, one of the beautiful campus I have ever seen. And since then, I'm working here. So for the introduction, phosphorus. So what is the role of phosphorus in plants? Phosphorus is among the essential macronutrient like nitrogen, like sulfur, calcium, magnesium. And it's required for the normal growth and development of the plants. Why it is required? Because it's an essential component for the key molecule like RNA, DNA, ATP, and it takes part in several energy-dependent reactions like signal transduction, electron transportation system, oxidative phosphorylation, dephosphorylation. So it does a lot of things, and that's why it's an essential macronutrient element. And it forms up to 0.3% of the plant's dry weight. So we can see that's how it is important. So phosphorus availability in general across the world, due to their less mobility of the phosphorus and it's less soluble in nature, it often makes complexes with iron and calcium and zinc, and that's why it's less available for the plant. So there is a phosphate starvation kind of condition in the world. In the industrialized and developed world, we used to have more phosphorus. We used extensively phosphorus-based fertilizer and animal manures in the field, and that often creates the problem of accumulation of phosphorus to the topsoil, and that leaches to the water bodies like river and lakes and cause eutrophication and algal blooms. So there are several examples, but two most important examples, one of the Chesapeake Bay in Maryland. And we can see here, this is the satellite image, and we can see the algal blooms, eutrophication sites. And this is this is for manure, and this is for commercial fertilizer, and these are the sites which has heavily contaminated with excess phosphorus. And these are the 180,000 kilogram every year phosphorus has been deposited. And that's why the phosphorus pollution is mainly due to the agriculture-based fertilizers. In Eastern Shore, it is 80% contributed to the agriculture. In Maryland, it's 50% contribution, and in watershed region, it's 58%. So it creates access problem in the environment. And second is the Okeechobee Lake in Florida. In the Florida upland ecosystem, the normal range of the phosphorus is 1900 to 3000 milligram per kilogram, and that is very high in the intensive agricultural areas. We can see this is a recent picture of 11 July 2016, and we can see the river of Lucy River, and we see the algal blooms, and this water quality is completely deteriorated. So this is a story is covered by National Geographic, a glimpse of the fertilized world. It's completely covered with the algal bloom. So how can we mitigate the adverse effect of phosphorus? We can do two things. We can increase the phosphate use efficiency of the plant so that there is a less need to put fertilizer. And second, we can formulate a strategy for phytoremediation of excess phosphorus. Phytoremediation is an efficient aesthetic green technology by which plants can accumulate phosphorus into the above ground part for phytoextraction. But the problem is, unlike the phosphate starvation response, there is no study for the excess phosphate response or to characterize plant responses under excess phosphate. Also, unlike the iron and zinc, we know that how iron creates toxicity, we know how zinc creates toxicity, but there is no information for phosphorus, that how when phosphorus increases, it causes toxicity. So in the present study, we have characterized comprehensively excess phosphate response in the plants. We take Arabidopsis thaliana as a model system, and we studied EPR. Let's see a little bit for phosphate starvation response, because it's help as a reference. So these are the soil particle, and this is the root hairs. From root hairs, plant used to take up nutrients and phosphorus, and then it goes to the inside the root, and these are the cell membrane which contains several transporter, phosphate transporter. So this is the phosphate uptake transporter, and then there are other transporter like loading, which is responsible for xylem loading, and that's why phosphorus mobilized from root to shoot through xylem loading. So root basically exert local phosphate response, local sensing, and shoot controls the systemic sensing of the phosphate. There are some typical responses in root, like root system architecture, root exudates. In shoot response, reduced photosynthetic activity, increased sugar content, increased anthocyanin biosynthesis, and phosphate recycling mobilization. So we designed an experiment, a concentration dependent experiment from -P to the excess phosphorus, and we keep increasing concentration to see the effect of phosphorus. So for this, we set up a hydroponic experiment in which a polypropylene mass has been used, 250 micron size. We put the seeds, sow the seeds on that mass, and we put inside the magenta boxes using the wedges, polycarbonate wedges, and the plant grows like that. So this is our strategy for giving the treatment. We have modified MS nutrient media, particularly to increase the phosphate content, and rest of the metal remain the same. We grow plants for 5 days in normal media.